Postsynaptic potentials are all about how the cell that is receiving the incoming information responds. So taking a look at this typical synapse, we know that a synapse is made up of an axon terminal, which will release neurotransmitter into this fluid-filled gap called the synaptic cleft, and that neurotransmitter will diffuse across the synaptic cleft and bind to receptors on the postsynaptic cell. The postsynaptic cell that's binding to the neurotransmitter is going to have a response, and that response is what's known as the postsynaptic potential. Postsynaptic potentials are the same thing as graded potentials. A graded potential is the way that the dendrites and the soma can receive incoming information. On the dendrites and the soma, there are chemically gated ion channels, and those chemically gated ion channels generate something called the graded potential, or in other words, the postsynaptic potential. A quick look at chemically gated ion channels that are located in the dendrites and the soma. Basically, they're closed. They're integral membrane proteins integrated into the phospholipid bilayer. They're normally closed, but when the neurotransmitter is released into the synapse, called the ligand here, that neurotransmitter will bind to the receptor, which is actually the channel. That is what opens the channel. Once the ion channel is opened, ions will cross it passively. And so if it was a sodium channel or a potassium channel, the direction of the movement depends on the concentration gradient of those ions. In the case of the neuromuscular junction, what happens is a motor neuron will release acetylcholine from the axon terminals, stimulating those muscle cells to contract. But when we start considering a postsynaptic potential, the postsynaptic cell could be a skeletal muscle cell, but it could be something different. It could be smooth muscle, it could be the heart, it could be a gland. And so there are other questions that we have to take into consideration when thinking about a postsynaptic potential. So I'd say that there are three main additional considerations that you have to take into account. One is, what type of chemically gated channel is being opened on the postsynaptic cell? In the case of the neuromuscular junction, we know it's the sodium-potassium channel, but that's a question mark in a postsynaptic cell, in a generic postsynaptic cell. We would also need to know how many neurons are synapsing with the postsynaptic cell, and how frequently are those presynaptic neurons releasing neurotransmitter. And so there are many additional questions. Let's first take a look at, what type of chemically gated channel is opened. If a neurotransmitter is released that opens a sodium and potassium channel on the dendrites and the soma, then knowing the conditions of the cell, knowing that the sodium concentration is high outside the cell, sodium concentration is much lower inside of the cell, and that potassium concentrations are higher inside the cell, lower outside the cell, we can predict what type of movement will occur. So if we open a sodium-potassium channel, what will happen is that sodium will passively move into the cell, and potassium will passively move out. Now the concentration gradient for sodium is much larger, and so you're going to have a lot more sodium moving in. And when we have a positively charged ion moving into the cell, it makes the interior of the cell more positive. It lessens the difference between the inside of the cell and the outside of the cell. So now instead of the resting membrane potential being at -70 millivolts, now we have what's called a graded potential, which is of variable strength, right? It depends on how much sodium comes in. But that sodium that comes in is going to diffuse towards the axon hillock, and it has the potential to depolarize the axon hillock to threshold. And if it does that, then the axon will generate an action potential. And because of that, when a neurotransmitter opens the sodium channels in the dendrites and soma, that's known as an excitatory postsynaptic potential. It's excitatory because there is a possibility that that could lead to an action potential by that depolarization moving into the axon. Here's another scenario. What if the neurotransmitter that was released into the synapse bound to chemically gated ion channels, and those ion channels were either potassium channels or chloride channels? In both of these cases, what will happen is the opposite of what we just saw. We know that potassium concentration inside of the cell is higher than the potassium concentration outside the cell. And so if a potassium channel opened due to neurotransmitter binding, then potassium is going to leave, a positive charge leaves. If a chloride channel opens, chloride concentrations are higher outside the cell than they are inside the cell, and so a negatively charged ion would come in. Either way, it's going to result in the inside of the cell becoming more negative, or what's known as a hyperpolarization. It's a hyperpolarization because if this cell is resting at negative 70 millivolts, when a positive charge leaves or a negative charge comes in, it becomes even more negative, like maybe negative 80, negative 90 millivolts. And because of that, that effect can spread down to the axon and make it harder for the axon to elicit an action potential. And that's what's known as an inhibitory postsynaptic potential. So that answers the question about what type of chemically gated channel is being opened on the postsynaptic cell. If a sodium channel is opened, that leads to a depolarization. That's an excitatory postsynaptic potential. Or if a potassium or chloride channel is opened, that will lead to a hyperpolarization, and that's known as an inhibitory postsynaptic potential. So now let's take a look at these two questions. How many neurons are synapsing with the postsynaptic cell, and how frequently are the presynaptic neurons releasing neurotransmitter? So one of those questions alludes to the fact that unlike the neuromuscular junction, when we're looking at a neuron as the postsynaptic cell, it's possible and likely that many neurons are synapsing with that cell. There could be thousands of neurons synapsing with that one postsynaptic cell. And so how does that postsynaptic cell know how to respond since it's receiving input from so many different places? It's kind of like this comic is showing us how some of the neurons are telling the postsynaptic cell to fire, some are telling it not to fire. And so what will happen is the neuron will put all of these messages together and pay attention to the sum of them. And that's known as summation. There's two different types of summation. One type of summation is called temporal summation, and temporal summation just has to do with how frequently the excitatory postsynaptic potential is being given. And so in this example, we can see that there was a subthreshold depolarization, which did not generate an action potential. The postsynaptic neuron returns to resting membrane potential, and then another subthreshold depolarization. And because they're subthreshold, neither of them lead to an action potential. The next graph shows us that if those two subthreshold depolarizations were given together closer in time, that they could add up to a threshold stimulus. And so subthreshold stimulus 1, before it returned to resting membrane potential, another depolarization rolled in, and because you're closer to threshold, it makes threshold. And this is what's known as temporal summation. Temporal summation is adding up depolarizations that are close together in time. Another type of summation is called spatial summation, and spatial summation is not using time as a variable. Instead, it's using different spaces. So here we can see one neuron synapsing with the postsynaptic cell. Here's a second neuron making a synapse. And if those two neurons delivered a depolarization to this cell at the same time, like they're showing us on the graph, they're showing us two possibly subthreshold depolarizations, they could add up to a threshold depolarization when given at the same time, just from two different spots. So to build to the story, what if these two neurons were giving opposite directions? What if one of the neurons was causing a depolarization in the postsynaptic cell, and the other neuron was causing a hyperpolarization in the postsynaptic cell, and those graded potentials roll down to the axon hillock? Let's take a look at the graph. So I can see a depolarizing postsynaptic potential, hyperpolarizing, so that's inhibitory postsynaptic potential. And when they're given at the same time, they basically cancel each other out. So this is a little bit like a math equation where the cell just pays attention to the sum total of what's going on. So summation gives us more information about these issues. It takes into consideration that the postsynaptic cell might have many neurons synapsing with it that are giving the same or different information, and the frequency of those, the release of neurotransmitter will also play a role in whether an action potential is generated. So, in summary, there are two different types of summation. There's temporal summation, that would be giving depolarization or hyperpolarizations frequently so that subthreshold messages add up to being threshold. And there's spatial summation. Subthreshold stimuli given at different locations could add up to a threshold stimulus.